This is lecture 28. We're starting a new topic. So, for the next 7 lectures, we'll discuss anabolic cellular signaling. And in this one, I'm going to go over a lot of content. I'm going to cover a lot of proteins. Don't panic if it seems overwhelming. I would be stunned if it didn't seem overwhelming. Just stay focused on the review questions. And we're going to gradually build the machine of anabolic signaling piece by piece, but I'll reference a bunch of those pieces today. And we'll keep going over the relevant parts in future lectures. So, again, just stay focused on the review questions for now as we move through today's lecture. But this is where we're headed. In the next 7 lectures, we'll cover what hypertrophy is, why it happens, how it happens, mTOR, which is the bulk of the how, and then we'll do some practical applications at the end of this 7 lecture series. So starting here, skeletal muscle might account for about half of your body mass, depending on the person. And that mass is filled, it's just like packed with proteins. And with muscle metabolism and protein turnover, you can think of these things as constant adaptation that's aimed at fitness, which means it's aimed at self-preservation and, if possible, flourishing. But we're not going to flourish if if our skeletal muscle is too big or too small. It has to be the third little bear. I think that's the just right porridge. It's been a long time since I've read Goldilocks. All I remember is Goldilocks is an asshole. It's like, if you've read it, sort of, she breaks into other people's houses, bears are people too, steals all their stuff and like eats their food. And like it was like hot. It doesn't matter. Um, Somehow that the lesson isn't one of criminality, it's about finding middle ground. And in the body, middle ground is achieved by protein turnover. Muscle can't be too big, can't be too small. It has to be just right. It has to fit the stresses of the world in which you're operating. So, muscle cells, they're constantly synthesizing and degrading their proteins. Hypertrophy is protein synthesis outpacing protein degradation. You're translating more protein than you're degrading, so your cells grow. If you degrade faster than you synthesize, that's atrophy. And I'm going to return to those terms at the end. And it's not just muscle, it's bones, it's tendons, it's ligaments. All your cells are doing this, but muscles are doing it all the time and a lot. Muscle is very sensitive to growth and decay, to hypertrophy and to atrophy. You're synthesizing protein over here, you're degrading it over there. That's protein turnover. And there's more than just actin and myosin in your skeletal muscle. Those are the proteins we think of, but structural proteins get deposited too. Titin, nebulin, myomesin M protein, alpha actinins, desmin. There's tons of these things. So I go to Uganda sometimes, and half of Africa's bird species live there. More than 10% of all the world's bird species. And most of them aren't exclusive to Uganda. They're just like regular old native. But skeletal muscle is similar, except that the birds are proteins. So, maybe 70%, ballpark figure, about 70% of all the proteins in the body are expressed in skeletal muscle. Not exclusively, but they are native there. And then some are exclusively native, like myosin heavy chain 4. So there's some exclusively native proteins. But the point is, skeletal muscle is a hub for protein. Like if the Magic School Bus went on safari, they'd see enormous protein diversity in skeletal muscle. But there are some core players that you see elevated with hypertrophy. Some primary proteins you see getting deposited. And again, actin and myosin, those are some of the important ones. They're just not the only proteins that matter. So that answers the what is hypertrophy B. We're getting into the why questions now. Why and how questions are different. Like, why is the sky blue? Or what the kids always ask. That implies purpose. What is the purpose of having a blue sky? Well, on the whatever day, the second day or something, God Almighty, let's say it's the eighth day, the Almighty decides to take his or her, its divine palette to the upper air, and she's thinking about colors at the time. Grandma's eyes were blue. She used to make the best cookies in the world. And so in honor of, Lord Granny, the sky is purpose. Those questions are pretty much impossible to answer. Um, why questions are how questions are much easier to answer. They're complicated, but they're way easier to answer because they're about mechanisms. How is the sky blue? Well, color perception is the interpretation of different wavelengths. Like some wavelengths we hear as sounds, right? Others we see as colors. Um, there's like heat up last night's leftovers, but the visible wavelengths in the sky, we see some more than others, and there are gases up there, nitrogen and oxygen. There are gases in the sky, and it makes the light waves scatter, and the blue wavelengths are pretty short, so that's what you end up seeing. There are now questions that are easier to answer because they're just mechanisms. Um, so that's if like a little kid in the backseat asks, if he asks how, he or she asks, how is the sky blue? You have a good answer. If the kid asks, why is the sky blue? You just berate them. Like, you need to think before you speak, like you're really disappointing me. Like tell the kid they're never going to amount to anything. But so why is exercise good for you? And why does lifting cause hypertrophy? I'll try to answer these, but the answer it's about fitness. Exercise is good for you because it's bad for you. We talked about this really early in the semester. In the moment that you're exercising, exercise is horrible for you. It's threatening your heart and circulation. It's threatening your bones, your muscles, your immune system, buffering capacities. Everything gets messy during exercise. And the bigger the mess, the greater the likelihood of harm. So exercise is really bad for you when you're doing it, but when it's all over and your body has a minute to recoup, it can reinforce its tissues. So the next time you subject it to that kind of a stress, it might be better prepared. It will have remodeled itself a little bit. So it's a matter of self-preservation. So look at like the pictures. There's two different exercisers here. The guy on the left, he's running so hard, he's creating disturbance, like contrails in the air behind him. And like, so he's putting himself with that amount of vigor, with that intensity, he's putting himself at a state of risk. Um, if he keeps this up, he's going to have a heart attack and some sprained stuff and whatever. He's putting his body at enormous risk. But compare that to the guy on the right, in his warm-up pants and his scrubs top. Um, there's no risk walking. I mean, he looks like a serial killer, so there's like risk to other people. But walking, he's not putting himself really at any risk here. No internal, no organ damage, no structural damage to his tissues, nothing like that. So comparing the two bodies, the contrail guy and the serial killer, which of their bodies is going to adapt to the exercise that they're doing? Not just tolerate it unchangingly and move on, but actually adapt to it. You obviously know the answer to this one. Long, mellow exercise like vacuuming or gardening, that stuff burns calories, but it doesn't cause adaptation. Um, so the why of adaptation, the purpose of adaptation is fitness. So continuing with the idea of fitness, as you know, it has nothing to do with like six pack abs or whatever. Fitness is compatibility with your environment. It's appropriateness or suitability with your environment and its stressors. So is your body, compatible with your daily tasks and threats and whatever? If so, it's fit, right? It fits the environment in which it lives. So in this diagram, it's like a Capricorn. It's a very binary. This is female, male. A Capricorn woman is compatible with an Aries man, it looks like. That's a match made in like astrological heaven, cosmos, whatever. Uh, they fit together, right? The Capricorn and the Aries. Same thing with your body and the world in which it resides, with all of its loads and its stressors, right? Subject your body to exercise, and you change its I don't know, perception of its environment. Like, okay, so let's say it's a weightlifter. The environment in which they live and operate is one that demands like weird, seemingly pointless heaving for, I don't know, an hour at a time. So the body will respond by enhancing the structural integrity of those tissues, of the heaving tissues. If you have to like if you have to like sing for your supper, or whatever, you have to like sing for food, your body will adapt so you can sing better. If you have to chase meat before eating it, your body will adapt to do that so you can be better equipped to capture and kill deer or I don't know, dogs or aardvarks, whatever it is that you have to eat. So no matter what stresses you apply to your body, its cells will attempt to become more compatible with those precise stresses. So that's the why. That's the best I can do. It gets a little bit whimsical, but the best I can do about why does hypertrophy happen. Moving on to the how questions, the mechanisms, the scattered wavelengths of muscle fist. For the most part, the mechanism of you know protein turnover, it's a bunch of cell signaling cascades. Now there's some steroid hormone activity in there as well, but in general, and steroids for the most part are bypassing these cell signaling cascades. But in general, most of your repair and your remodeling, this includes growth and other adaptations. Most of this stuff is the result of signaling cascades. So the picture here below, the picture you can see, that's how a signaling cascade works. Now the drawings are as lifelike and like anatomically and chemically accurate as a Peanuts character, right? It's missing a level of detail that would make it realistic, but it illustrates the basics of cell signaling. And the bulk of signaling is a process called phosphorylation. We went over this in the last lecture. And the best illustration I can give for a phosphorylation cascade is these two people who are flashing gang signs at each other, but they're also passing a baton, sort of, kind of. They're sort of passing a baton to each other. And that's the whole point. It's a relay race. Let's say it's a 4x1, which means there are four runners and each one runs 100 meters. The starter pistol goes off. That activates the first runner. The rest of the runners are alive, right? They're present, they're on the track, but they're inactive. They're just hanging out, dormant, waiting until the first runner passes the baton. That activates the second runner. So the first runner at that point reverts to a dormant phase. And then the second runner is now active. And the second runner passes the baton to the third runner. That activates the previously dormant third runner to do the same thing. She activates the fourth runner, and then the fourth runner, crosses the finish line. And when that anchor runner crosses the finish line, they're given the trophy, right? In this case, it's a pun. It's hypertrophy, like hypertrophy. That's a change in genetic expression. Hypertrophy is. It's a change in genetic expression, which results in the synthesis of more proteins. Um, and so, let's go to the ribosome and cook us up some meat. So there, the baton would be the phosphate. The phosphate in a phosphorylation cascade activates things. So to answer the how question, it's mostly cell signaling. So take insulin, for example. One of insulin's many responsibilities, the primary, I guess, responsibility is to get sugar into cells from your blood, where it holds the title of blood glucose, into the inside of the cell, where it's either consumed in glycolysis or it's used as fodder for glycogen synthesis. But insulin doesn't just barge into the cell, right? Find a GLUT4 protein, like, oh, there you are, like, you can't hide from me, and then just like shove it, like grab it and escort it to the surface. That's not how insulin works. Insulin binds to a receptor on that cell's surface, which triggers a thing, which triggers a thing, which triggers a thing, which triggers a thing. It's a big relay race. The baton activates one thing, which activates another thing, which activates some other thing. So in the cell, again, the baton is a phosphate, and generally almost always a phosphate. And these are phosphorylation cascades. So phosphorylation isn't the only form of signal transduction. It's just the main one for protein turnover. So what can the starter pistol be for to get this 4x1, this relay race started? Um, like we know there's a cascade that's going to gush into the cell toward the interior, aimed at the nucleus or the ribosome or some protein somewhere or whatever. But what starts the cascade, a few obvious possibilities would be epinephrine, insulin, and growth hormone, what you see here. It could be any hormone made from a string of amino acids. This is just a few of them, a few examples: an amine hormone, a peptide hormone, and a protein hormone. Or it could be an icosanoid, right? It could be like prostaglandins are going to bind to a cell surface receptor and send a signal into the cell. Now, we've seen this image before, but let's take another look because it shows the signaling cascade of epinephrine in skeletal muscle or glucagon in the liver. And let's say this is a muscle cell. So we're looking at epinephrine. So at the top of the screen, that's outside of the cell. Then there's a phospholipid bilayer. That's the membrane surrounding a cell that has a bunch of lipids in it, and some cholesterol in there. Embedded within that membrane, within that phospholipid bilayer, is a beta adrenergic receptor. That's what your epinephrine is going to bind to, your adrenaline or epinephrine is going to bind to. And those are just two words for the same thing. So in this case, that fires a starter pistol, right? That initiates the relay race. And in this case, the relay race begins with the it ends, right? So it begins with that, but it ends with the chopping of a glucose chunk, a little chunk of glucose from glycogen off of your glycogen. And that's a G1P, a glucose 1-phosphate, a glucose with a phosphate attached to that one carbon. And so this is glycogenolysis signaling, glycogen, lysis, glycogenolysis. This part of the cascade isn't nucleic, right? Its finish line is at the cytosolic carbs, the carbohydrate, the stored carbohydrate. So moving away from carbs and getting into fat, going from glycogenolysis to lipolysis, lipid lysis, lipolysis. Both of these can be activated with the same starter pistol, right? It's the same start to the signaling cascade. It just ends at different spots. So this is still PKA signaling, but what PKA is phosphorylating is different. Here in a fat cell, an adipocyte, it's like perilipin. Perilipin gets phosphorylated. That's the coat on the lipid droplet, which is concealing a bunch of triglycerides. So the picture of the creepy man would be an adipocyte, right? A fat cell. And the like predatorial trench coat, that would be perilipin. And what's being exposed is triglycerides. And you can see that in a less creepy form in the diagram to the left. And you can also see hormone-sensitive lipase getting phosphorylated, also by PKA, and that's performing lipolysis on the newly exposed fatty acids. Now, lipolysis is a little bit more complicated than that. You don't see adipose triglyceride lipase here or monoacylglycerol lipase. You just see the middle sibling hormone-sensitive lipase. But adipose triglyceride lipase would go first. HSL, hormone-sensitive lipase, that goes second. Monoacylglycerol lipase, that goes third, that goes last. Now you have three fatty acids that used to be all conjoined, freed from one another to go roam freely and be burned. Here's a cleaner picture of the same thing, but it gets rid of perlepen and adds insulin to the signaling cascade. So the reason you have to go through lipolysis at all is that a whole triglyceride, the glycerol, and the three fatty acids, that big log is just too big for the fireplace. You have to split it up. You have to take a hatchet to it. And that's what those lipases are doing. That's what those enzymes are doing. Again, in this diagram, you don't see adipose triglyceride lipase. You just see hormone-sensitive lipase and monoacylglycerol lipase. That one's in this diagram too. But it's a nice diagram anyway. It shows the big steps in chopping up the firewood into burnable hunks of lipid firewood. And we'll talk about insulin's role in all of this. But before we get to insulin itself, let's look at AMP and cyclic AMP. Cyclic AMP is the thing that activates PKA. And PKA, protein kinase A, is the thing that phosphorylates all your worker bees, the enzymes responsible for the actual labor of lysing. PKA is their boss. And to turn on that boss, to turn on PKA, you attach cyclic AMP to it, four of them. Four cyclic AMPs bind to PKA, and now PKA is turned on. But it's not turned on by regular old AMP. Regular old AMP and PKA are not as companionable as an Aries and a Capricorn, right? You need cyclic AMP to pair with it, to pair with the PKA. And we make our cyclic AMPs from ATP. That's what adenylate or adenylyl cyclase does. That AC, that's adenylate cyclase. That's the enzyme that epinephrine and glucagon are activating. They bind to their cell surface receptors, which is a G protein coupled receptor. A subunit of that G protein binds to adenylate cyclase. That activates it. You don't need to know that. But adenylate cyclase gets activated, and that converts ATP into cyclic AMP, which binds to PKA, which then does PKA, then does its phosphorylating. It starts phosphorylating anabolic things. But if you convert that cyclic AMP to regular AMP using PDE, phosphodiesterase, PDE, you inhibit PKA's activation. That's the mechanism. That's how insulin obstructs lipolysis in the short term. It activates PDE. This is a closer look at phosphodiesterase activity. Adenylyl or adenylate cyclase is converting ATP into cyclic AMP, and PDE is converting it to AMP. And AMP can't activate PKA, so no lysing happens. Just like how insulin doesn't translocate GLUT4 all by itself, it does so by way of a signaling cascade. That same cascade, the primary players being PI3K and PKB, turn on PDE. So this slide has the narrative version of those events. PI3K and PKB can activate phosphodiesterase. So these are just the steps here on the screen. And this is another look at the same phenomenon. This one is a really good illustration of phosphodiesterase, PDE, as a liaison that relays communication between PKB and PKA. So pay attention to this image, this slide, and you really know everything here. On the left, that's a diagram of beta adrenergic receptor activity, that cascade, adenylate cyclase, break ATP down into cyclic AMP, bind four of those little guys to PKA. PKA gets activated, and you see it phosphorylating both perilipin and hormone-sensitive lipase. And now you're mobilizing your fatty acids. On the right side, though, there are a few blobs worth noting. IRS, that's insulin receptor substrate. It helps transmit signals from the receptor to the cascade. PDK, that's phosphoinositide dependent kinase. And PIP3 is phosphatidylinositol triphosphate, 3 phosphates. These will all come up later. So it's just helpful to recognize those names, to become more and more familiar with those names. But you see the interaction between PKB and technically PI3K to PDE also, and how that can stop beta adrenergic signaling. So here's a closer look at insulin signaling specifically. The root word for for where insulin comes from, the pancreas, is pancreas, which means all, like a panoramic, right? Or a panacea, it means all. Creas, flesh. So it gets everywhere. Insulin gets at the stuff that comes out of the pancreas, the stuff that comes out of the all flesh, gets everywhere. So how does insulin get everywhere? It's an endocrine hormone. So it gets out of the blood, out into the blood, right? And then, it's out in the, it plays for a while, circulates for a while in the blood, and then it leaves circulation and binds to its receptors that live wherever, that live all over the place on whatever target tissue. And once insulin binds, it initiates PI3K, PKB signaling. Now, downstream from PKB, tons of stuff happens. So PKB is sort of a pan kinase enzyme, very diverse functions, but its function, it's all really anabolic. There's no catabolic stuff here. PKB is just anabolism, anabolism, anabolism. So it's very anabolism. Certainly not helpful if you have, cancer. But if you're looking to grow, it is very helpful. So just focusing on PKB, we see an upstream input. We'll talk about those steps soon, but there's a path to turn on PKB, and then there are multiple arrows headed out. And PKB, in a lot of these diagrams, PKB is written as AKT. That's the AKT you're looking at here. It's the same thing. That's PKB. That's protein kinase B. But it has a lot of targets. There's a lot of things that PKB phosphorylates. And this is a look at one of those possibilities, one of those targets, mTOR. Now, mTOR is just another hub in these signaling cascades. PKB is a major hub. mTOR is another major hub, and it's another kinase. It's an enzyme that phosphorylates stuff. Downstream from mTOR's activity, actual protein synthesis happens. mTOR is pretty close to the finish line. mTOR isn't the finish line. It's pretty close to the finish line. But you can see what PKB is doing here. Okay, it's inhibiting tuberin. TSC1/2 tuberous sclerosis complex. Technically, TSC2 is tuberin. And technically, there's also a RHEB, Ross homolog enriched in brain, that should be in this diagram between tuberin and mTOR. We'll talk about that soon. I'll show it to you soon. Um, and even here, when you see P70S6K, that also isn't a direct shot to synthesis, to protein synthesis, but you're at the last leg of the race. That's another kinase, and it's really close to the finish line, that one, P70S6K, really close to the end. Um, Now, remember glycogen synthase and glycogen phosphorylase with PKA signaling? They both get phosphorylated, but that turns off glycogen synthase and it turns on glycogen phosphorylase. A similar phenomenon happens here. mTOR, the kinase, phosphorylates both P70S6K and 4EBP1, which turns on the former and turns off the latter. So here you see an arrow pointing to um, uh, PKB, or just one arrow, AKT, right, is listed here, but that's PKB. One arrow pointing in and turning it on. That's PDK activating PKB. Technically, there are PI3K independent pathways that activate it, but this is the primary way, the primary mode of PKB activation is through PI3K. And then once PKB is on, you see a bunch of arrows going out, a bunch of influences, PKB's activities. They're abundant. And if we upgrade our image, right, you see a lot more activity from PKB. Now, some of it you should start to recognize, tuberin and mTOR on the left, uh, P70S6K, 4EBP1. On the top at the cell surface, you see cytokines and growth factors, a bunch of stuff that triggers PKB, going through that, PI3K signaling cascade. We'll start outlining more and more of this map as we move forward, but plenty of this content we're going to skip over. So don't feel too overwhelmed by the number of players in this on that team there. But let's look at some major domains of PKB signaling. There are only a few things here that you need to know, and I'm going to cover it broadly, but the only things that you have to know, that I'm going to hold you accountable for, are FOXO, AS160, TSC2, which we've already talked about, that's tuberin, uh, Pras40, and GSK3. And with each of these, I'm going to go over it again and again in clearer and more simple detail. So don't write down anything I say on this slide. Just hear me passively. Don't panic if none of it sticks. Everything that needs to stick, we're going to go over again and again here. I just want to give you the overall picture. And a lot of this you're not going to be held accountable for. So among PKB's diverse functions, uh, there's anabolism and proliferation, cell survival, glucose uptake, angiogenesis. These are all in PKB's like wheelhouse. And so let's begin with angiogenesis. That's the formation of new blood vessels from old ones, branches from already existing roadways. You see ENOS in the picture, endothelial nitric oxide synthase. PKB phosphorylates that. It turns it on. PKB turns on ENOS. So if you inhibit PKB, you turn off that activation of enos. Upstream from PKB in this case would be vascular endothelial growth factor, VEGF, that activates PI3K. Then PKB gets activated. From PKB, you activate enos. And then from there, you're getting closer to the finish line of angiogenesis. Okay, let's move on to survival. Um, I mean, like all of these pathways are critical for like biology's ability to keep on living. That's not what I'm talking about. What I'm talking about here is cell survival specifically. And in this case, PKB is shutting off programmed cell death. This is a big piece of homeostasis and very general regulation. The body has no shortage. So apoptosis, right, is like programmed cell death. There's just no shortage of pro-apoptotic things. And PKB, what PKB is doing is inhibiting a lot of those things. So CASP9, that's one of them. It's a caspase, cysteine aspartic protease, caspase 9, which implies there's like an 8 and a 7 and a 6, like all the way to 1. And in the other direction too, there's more than 10 of these things. There's a lot of these protease enzymes. And they're involved in inflammation and apoptosis, and PKB is shutting that stuff off. BAD, right? BAD, that's another apoptotic protein that PKB phosphorylates. So BAD, it's BLC2 associated agonist of cell death. So B, blah, 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 whatever, agonist of death, right? That's bad. And it's inhibited by PKB. So PKB is inhibiting that thing too. And this isn't the full extent of PKB's interaction with cell death, but you get the idea. PKB is stopping it by phosphorylating and inhibiting stuff that would otherwise promote cell death. MDM2, mouse double minute 2 homolog. You really don't need to know this one. Part of its function, though, is the P53 signaling cascade. It represses P53's activity, and P53 is a tumor suppressor. Under normal conditions, the half-life is under 20 minutes. It's pretty short. And its levels are generally really low because MDM2 mediates its degradation. It's a really weird relationship between those. So the transcription of MDM2 is actually induced by P53, but P53 is inactivated by MDM2. So P53 is like, it's like a masochist that's hiring MDM2 as his dominatrix. And then when PKB phosphorylates MDM2, it prevents its self-ubiquitination, so it promotes it. You see more degradation and negative regulation of P53, so you see less apoptosis. But P53, it's really important. It's the quote, the guardian of the genome. It's always called that, named for its role in tumor suppression. And the name, whatever, it's a protein of 53 kilodaltons, P53. You don't need to know any of that, but it's on the slide. So that's, if you're curious, then I hope that satisfies the appetite. In the glucose uptake section, this one is worth knowing, and we're going to cover it more, but you see AS160, AKT substrate of 160 kilodaltons. If that sounds gibberishy, if that name, the original name, its biological birth certificate was TBC1D4. So it's like when astronomers are naming like distant stars and planets by their celestial coordinates, and it just reads like serial numbers. The conventions of chemistry and biological naming, the nomenclature, astronomy, all these fields, it's kind of boring and calculated. And although I would argue it's preferable to naming the Kentucky Derby, those horse names awful. But in the case of AS160, it's not that bad of a name. AKT substrate of 160 kilodaltons. PKB phosphorylates that thing. PKB phosphorylates AS160, and that inhibits it. And when AS160 is inhibited, that withdraws its inhibition of GLUT4. So GLUT4 can translocate, and you can get sugar into the cell. That glucose transporter, we can move sugar into the cell. You have to inhibit AS160 to do that. Mess up regulation here, and you'll find yourself, hyperglycemic. And the last thing on, other than FOXO, but that I have a slide, an entirely separate slide for that. Uh, and it'll be a big topic in future lectures, FOXO will. But otherwise, the last thing is P27. PKB inhibits this, obviously by phosphorylating it, and it would otherwise have anti-proliferative effects. So that's why you see this little protein in the proliferation column, and you don't need to know it. But FOXO, again, we're going to talk about this more and more as this lecture series, these 7 lectures on anabolic signaling continue. FOXO is one of the pathways that people focus on in studying cancer, but metabolism and autophagy. Fork head box O, that's what it stands for. There's lots of FOX transcription factors. These are proteins that regulate gene expression. But with fork head box O, with this one, we see PKB directly inhibiting it by phosphorylation, obviously phosphorylation. And AMPK, something we're going to talk about a lot in the future, that has the opposite effect on it, activating FOXO. But again, a topic for future lectures. So let's take a step back from the how mechanisms. They can get heavy quickly. There's just a lot of vocabulary. And let's do a why question. So why is cell signaling important? So in this picture, we have an angry cloud, right, blowing at you. And that's like environmental stresses. Let's say that is environmental stress. And, you will read about like specific stresses on a weather app, right? Storm tracker or whatever. Here's the typhoon that's headed your way. When you see that, you decide to bring an umbrella, wear a raincoat, cancel your picnic or whatever it is. You respond to that information, to that environmental status update by changing your behavior. Your cells do that too. They need to. Your cells need to know what the environment is like so they can also change their behavior to be more appropriate or compatible to the conditions outside of themselves. Um, but they're not doing it with a smartphone, right? There are sensors on the cell surface. They're like wind chimes on a porch that relay messages into the interior. Oh, the wind's blowing strong today, whatever. Those messages that get relayed, they contain information about chemical and mechanical environments. And then once the cell receives that information, it'll change its behavior to better tolerate or accommodate those stresses. So consider the perspective of upstream and downstream, just these concepts. Now, the origin of the stream, this is an initiating stimulus, right? It's the starter pistol that commences the race. In the body, maybe you're exercising or you're eating, right? A dog barks at you. Whatever happens, that initiates a cascade of events. Downstream from that, you see some hormones being released. Downstream from that, those hormones are binding to their receptors. Downstream from that, you have a signal transduction, a cell signaling cascade that gets started. Some phosphorylation is happening. Follow the stream to its end, and you get lipolysis or glycogenolysis or GLUT4 translocation or changes in genetic expression, something, right? That is the end of that race. So upstream and downstream, these are both relative, and they reference the sequence of cellular events. So let's say the stimulus that gets everything started is you eating a hamburger. The white bread is a really high glycemic carbohydrate, so you raise your blood glucose in a hurry. That stimulates the release of insulin. Downstream from that, insulin binds to its receptor. Downstream from that, it activates its PI3K, PKB cascade, downstream, downstream, downstream. It just keeps going until it crosses the finish line. That burger also has meat in it, and meat has a bunch of amino acids, and there's some important amino acids here like leucine and arginine and methionine and lysine and glutamine. And the cell does a really good job of recognizing those amino acids, more than some of the other ones. Um, and so those amino acids are going to help initiate their own signaling cascades through the gator complexes, which we'll talk about later. If the initiating stimulus is a set of squats, that causes tissue disruption. Downstream from that, we get an inflammatory response. Downstream from that, inflammatory compounds bind to their receptors. Downstream from that, we have a signaling cascade and so on and on and on and on. So you get the idea of upstream and downstream. So let's look at a broader picture of cell signaling. This is a cell, uh, lots of cell surface receptors on it. The thick orange line is your phospholipid bilayer around the whole cell. That's what the cell surface receptors are embedded in. And all sorts of stuff can bind to the cell. And you can start to recognize a bunch of stuff here. PI3K, PKB, that's there. Adenylyl cyclase, PKA, that's there. You see some caspases and bad GSK, glycogen synthase kinase, that's in there. P27, P53 make an appearance. So there's lots of stuff you'll start to recognize, and we'll really work through a lot of this map in the coming lectures. But let's start on this side of it. JAK-STAT signaling. JAK, that's Janus kinase. STAT is signal transducer activator of transcription. We've seen this signaling cascade before with growth hormone, but lots of things signal through this pathway. We're not really going to get into it. We're going to focus on MAPK and PI3K, PKB, mTOR. But downstream, the effects are pretty diverse in this signaling cascade, including a lot of immune system regulation. Up here at the top here, we see PI3K, PKB, that signaling cascade. A lot of stuff, tons of stuff will activate this. And we'll talk more about it in the lectures to come. But, IGF, insulin, epidermal growth factor, transforming growth factor, vascular endothelial growth factor, VEGF, we just mentioned that one. Fibroblast growth factor, lots of growth factors. Factors are just proteins, and proteins that transmit messages do so through cell signaling, right? On the cell surface is the receptors that they'll bind to. In this case, RTK stands for receptor tyrosine kinase, and then GPCR, that's a G protein coupled receptor. Just different classes of receptors embedded in a phospholipid bilayer. Okay, this one is showing integrins. We're really going to get into integrins soon. This is mechanical stimulation. So this is probably more important to pay attention to this slide, just so you remember some of this stuff. But integrins integrate the cell, the inside of the cell, and the extracellular matrix, the ECM, extracellular matrix. That is the space right outside of the cell. There's a bunch of collagen and proteoglycans and glycoproteins and elastin. There's tons of stuff out there. And integrins are relaying the mechanical environment, loads and stresses to the interior of the cell from the extracellular matrix. And then notice FAK there, that's focal adhesion kinase, another kinase. It's a phosphorylating enzyme that gets activated by integrins. And then that message gets relayed all over the place inside of the cell by FAK. And that's mechanotransduction. Mechanotransduction, which just means mechanical signal transduction, mechanotransduction, as opposed to chemical. Over here, this is Wnt, W-N-T, but it's pronounced Wnt. It's another protein. It binds to a receptor called frizzled. When a Wnt protein binds to it, it transmits the signal to disheveled. And there are some roles in carcinogenesis and embryonic development. But for the context of this lecture, there's mTOR interaction with GSK, with GSK3. But you do see a relationship with autophagy, but more interestingly, when Wnt protein, more interestingly for us, for the context of this lecture, when Wnt proteins bind to frizzled, they're going to inhibit GSK3. GSK3 keeps tuberin phosphorylated, keeps it on. So if you activate tuberin, you're deactivating mTOR, right? If you deactivate tuberin, you can turn on mTOR. So Wnt proteins have a role in anabolism. Moving on to Hedgehog, you don't need to know this one. There are a few ligands for this pathway. Ligand is just a thing that binds to something. But the ligands here are things like desert hedgehog and Indian hedgehog and sonic hedgehog. That's the name of one of them. Mostly it's embryonic roles, teeth, stuff like that. But in the adult, there are also some roles that promote proliferation of stem cells. All right, so now we're looking at apoptotic pathways, apoptosis. Again, this is programmed cell death. And in the balance of anabolism and catabolism, there's so much interaction in these pathways that you can tinker with. But again, homing in on the center of the signaling so we don't lose our focus. PI3K and PKB, AKT here, but again, that's PKB. It's the same thing. This really is the primary route that anabolism takes. It's the major highway for hypertrophy, for anabolism. And there are lots of entrances and exits for it. Now, downstream from PKB, AKT, or PKB, again, same thing, you see inhibition of tuberin. And inhibition of FOXO and inhibition of GSK3. If you inhibit FOXO, you minimize atrophy. We'll talk more about this in the future. If you block tuberin, you shut off the thing that's shutting off the thing that's turning on mTOR. Now, REB, RHEB, again, that's ROS homolog enriched in brain. You don't need to know what it stands for, but that's an activator of mTOR. And REB is inhibited by tuberin. And we're going to talk more about this in the future, so don't worry if you don't catch it all right here. But PKB inhibits tuberin, which was inhibiting REB, but now it's not because of PKB. So REB is now free to activate the next step in the mTOR cascade, mTOR itself, right? So two negatives make a positive here. And then you can also see PKB inhibiting GSK3 here. If the double negative of PKB's effect on tuberin wasn't confusing enough, you now have PKB shutting off GSK, which was turning on tuberin, which was shutting off REB, which was activating mTOR, right? So as we keep moving forward, you'll see anabolism, hypertrophy, growth, remodeling of protein. You'll see it looking more and more like a Rube Goldberg machine, like just this really complicated machinery that seems like it could be, and should be much simpler. It's like that old board game Mousetrap, if you have any idea what that is. That's sort of what cell signaling starts to look like. At the finish line for these signaling cascades, often it's nucleic, it's the nucleus, start at the cell surface and end with some, genetic regulation, but it doesn't have to be that, it could be moving GLUT4 translocation, or it could be breaking up glycogen, lysing glycogen, glycogenolysis, or lipolysis, or 1,000 other intracellular, but not nucleic functions. But the cell signaling is somewhere in the cell. It has some function in the cell. Now, Protein synthesis is genetic. It's genetic expression. That's what anabolism really is. It's genetic expression. And transcription comes first. Translation comes second. Transcription, that happens in the nucleus. You make some mRNA and then you send it out of the nucleus and to the ribosome. And that's where it can be read. It can be translated. So the ribosome is basically a kitchen. And the mRNA is a recipe. And that recipe sent by the nucleus is the instructions to cook up a protein. And so translation is how we're synthesizing proteins. Earlier, when I said protein turnover is the balance of synthesis and degradation, translation is synthesis. Are we translating more protein than we're losing? That's hypertrophy, that's growth. If you're translating less than you're degrading, if the ubiquitin proteasome system and the lysosome are eating up your proteins faster than you can cook them, that's atrophy. And we're going to talk about atrophy soon in the coming lectures. Um, and there are behaviors, there are things you can do. There's stuff you can eat, there's stuff you can do in the gym, there's supplements you can take, there's stuff you can do that will affect that balance. But that brings us to our first checkpoint. It is a reasonable time to reflect on what we've been talking about and telegraph the past that's coming. So what is mTOR? Technically, mTOR is a kinase. It's an enzyme that phosphorylates stuff, attaches phosphates to things. But mTOR also refers to the whole signaling cascade. mTOR signaling, PI3K, PKB, mTOR, and then on from there to P70S6K and so on. That's a whole signaling cascade. Uh, and lots of stuff serves as your cell's weather app. Mechanical loading does it, chemical signals, lots of stuff. That stuff informs your cellular machinery, what the environment is like outside of its walls, outside of its, phospholipid bilayer, outside of itself. And that information changes your cell's behavior. If it turns on mTOR, your cells are then growing. Now, the remaining four questions here, that's what we're going to be talking about in the next lecture. What is an mTOR complex? What complexes are there? What are the steps of mTOR signaling? And what does that signaling accomplish? All that's coming soon. But this is where we'll end with our review questions. Really focus on this stuff. I provided a lot more information than you need to know. But this is foreshadowing what's to come. So a lot of the vocabulary that was in today's lecture is going to be repeating in the coming lectures. But I will see you in the second of 7 in this series on anabolism.